is a code and there is information. The improbable order or arrangement of amino acids and proteins does contain information, because the order is arranged to produce a specific effect. It results in, and is necessary for, function in the cell. In this particular example, the bases AUG on the mRNA link up with and express a specific amino acid. The amino acid expressed by AUG is called methionine. There are actually tables that show which codons, or sets of 3 bases on mRNA, express which amino acids. The base sequence GCA, for example, expresses the amino acid alanine. So this amino acid chain would be methionine and then alanine. Now remember, the amino acids must wind up in precisely the correct order for a protein to form and for life to exist. If you get the wrong order, there is no life. As stated earlier, a relatively short protein would be comprised of about 150 amino acids in precisely the correct order. Many proteins require thousands of properly sequenced amino acids. Think about that. That means that when the bases in the DNA molecule on the double helix are formed, since the arrangement of those bases will determine what amino acids are eventually expressed, those bases must be formed in view of eventually, down the line, correctly expressing every 3 base codon that will link up with every amino acid. And the DNA bases must express them in precisely the correct order, or there will be no life. And here's what's most important on this point. The linking of codons, or 3 base sequences, with specific amino acids is not determined by physical properties or by chemical reactions. Chemical reactions do not determine that GCA will link up with the amino acid alanine. Notice the distance that exists between GCA, its anticodon pair CGU, and the amino acid expressed by GCA. They are at opposite ends of tRNA. That means that chemical reactions between the codon GCA and the amino acid it expresses, alanine, cannot account for their assignment. No, there is something else that determines the assignment, something that transcends the chemical properties of these things. This is also true of the arrangement of the bases on the DNA molecule. Why are the bases in DNA arranged in the precise orders which enable them to achieve functional sequence? The orders in which they are arranged are highly improbable. What accounts for it? Is it a result of chemical attraction? No, it's not. One cannot logically argue that chemical bonds between the bases in DNA determine their proper sequencing or account for the unique arrangement we see in DNA. That argument will not work because there are no bonds at all between the 4 bases on the longitudinal north-south axis of the DNA molecule. If we look at this picture, focus on the second set of bases on the left side, A, C, G, and T. Those 4 bases are across from TGCA. Now, as we look at these bases, notice that there are bonds running horizontally between A and T, and between C and G. These horizontal bonds link adenine and thymine, and cytosine and guanine. But notice that as we look vertically, or longitudinally, north-south, there are no bonds at all between A and C and G, and G and T, etc. Since there aren't any bonds along this critical information-bearing axis of the molecule, north and south, chemical reactions cannot explain why A would be followed by C, and C followed by G, etc., in the extremely improbable and non-repeating order we find in DNA. Since there aren't any bonds there, one could not say that guanine is chemically more likely to follow cytosine, etc. No, something else is arranging the order, and the proper order must be achieved for life to exist and function. Moreover, if we look again at the letters A, C, G, T, notice how each letter or chemical attaches to the backbone of the molecule itself. The same type of bond occurs with all 4 chemicals. There is no difference. The same type of bond exists between the base and the backbone, regardless of which base attaches. When A links to the molecule, or when C, G, or T does, the same bond is present. There is no reason, therefore, chemically speaking, why one order of chemicals would form rather than another. All orders are chemically equivalent. Chemical properties, therefore, do not account for the unique, specific, and highly improbable arrangement of the bases in DNA. Something else accounts for it. The arrangement of the bases in DNA and the arrangement of the amino acids in proteins cannot be explained by chemical properties or chemical reactions. No, intelligence, a supreme intelligence, is the only explanation for why the bases in DNA and the amino acids in proteins are arranged in a way that transcends the materials involved to achieve specific effects. For what we have in every single protein is an extremely improbable arrangement of amino acids. It's more improbable than finding a single marked atom via a blind search out of all the atoms in the galaxy. This arrangement comes from a prior extremely improbable arrangement of the nucleotide bases in DNA. These extremely improbable arrangements are all organized to produce a specific effect.
Come with me now.